And welcome back to Microbial Growth Control. And will be our final video, and we will talk about how we use chemicals to control microbes in our environment. Okay. Again, we'll talk about antimicrobials and chemotherapy and antibiotics and all that sort of thing after this unit and after this chapter. Okay. And we'll get into how those systems manipulate the biology and biochemistry of the microbes. Right now, we are doing the same thing with chemicals here, but these will not be in our body. These will be on the outside of our body. All right. So you've already been using this every day, right? Hand sanitizer, or maybe every day in 2020 at least. Before you get your blood drawn or a immunization, the nurse will rub some isopropyl alcohol on you, usually in a pre-sealed swab now, rather than the bottle. But you might still get this bottle here and maybe sterilize some, nails equipment or the like, right? As we sterilize things at home. All right, so alcohols, how they work are they are going to denature proteins very quickly. Okay? The protein then is going to fold and get all funky and will no longer have an active site. And as I said in the lab, we disrupt these membranes, right? And this is why decolorizer works. We're going to disrupt some membranes and get rid of the color and take away that small layer of dye in gram-negative cells. Okay, so we're going to disrupt membranes, denature protein. Surfactants, okay, surfactants are these long molecules, okay, with an hydrophobic tail here. All right, they have a hydrophobic tail and they have a hydrophilic head. So they're very similar to the phospholipids that we use for the cell membrane. Okay, but in this case, we can take away hydrophobic things on our hands, like grease. Okay, soap will help against the grease, and we can also take away things like salts and other normal debris using our hydrophilic head, and that's why soap works so well. It's just going to carry things away with it using the charge over here and the fact that this is not charged over here. And soap is, remember, a degermer. Okay, these two are both degerming. Okay, they are not antiseptic. They're degerming. Just to hammer it home one more time. Make sure you remember proper hand washing and what happens when you don't hand wash in an healthcare environment, right? Or at least be aware of the shortcomings in hand washing that are inherent in it, right? Physical obstructions like nails and everything get in the way. All right. And so finally, we come to the end of our PowerPoint where we can actually see the effectiveness of our antiseptics and our disinfectants. Okay, we can put soap on a disc, we can put antibiotics on a disc, and we can put that on our nutrient agar. Okay, and we can discuss how much this affects the bacteria. Okay, we can see, oh wow, look at that. That disc is affecting that bacteria so much that it does not grow in this whole circle. Okay, just from that one molecule. Same with this one. This one's not as effective to this microbe. It might be more effective to another microbe. It's not here. On this one, the same antibiotics are used, and what did a lot of killing or disrupting of bacteria here ended up not doing a thing over here. It was like the cell did not recognize it, the cell metabolized it, one of the things, right? Those bacteria are not affected. They are resistant to this, okay? Meanwhile, this one is susceptible. So resistant, resist, and susceptible. Okay. All right. And so we'll do this in the lab. We'll actually get to do this. We're going to do it both ways. We're going to put some disinfectants on some discs, and we get to do some pre-made antibiotic ones. And we get to see what your case study looks like in the lab based on some antibiotics. So in your case study, you can actually determine what you might be able to prescribe to help your individual patient. Okay, and if my antibiotic that I prescribed looked like this, I would know why his symptoms progressed. Okay, you can't do this overnight, right? Like, or this has to happen overnight. So you already have had to have prescribed an individual with a rapid infection with something. Okay, you can't wait. Okay, so when we say that it didn't work, it's not that the doctor didn't do their job. It's that this needed to grow overnight. We needed to do it. So if in the morning, though, you discover your antibiotic that you did prescribe because it was a general antibiotic, you would see that you need to change it quickly, and you would move then to this guy. All right, so that's the implications of this test. It's not to memorize what a Kirby Bauer one looks like or to memorize the concentrations of these guys. It's to say, hey, look, I want to give this to my patient. Okay, and with that, we are done with our microbial growth control and our second unit of lectures.